September 3rd, 1939. The Radcliffe Infirmary. Oxford, England. The air inside the surgical ward was heavy, sterile, and permeated by the faint, inescapable scent of iodine and fear. Private George "Geordie" Miller, a young infantryman who had only just left his job in a Durham shipyard, lay pale and still on a narrow iron bed. The declaration of war against Germany had swept him up in its fury, but it was not a German bullet that threatened his life. It was a careless accident during basic training. A training rifle had fallen, crushing his foot. The immediate injury was bad enough, but it was the aftermath that truly terrified the nurses. The wound, a jagged, deep gash, was turning an angry, sickly green. Staphylococcus bacteria, the invisible enemy, had moved into the tissue. The standard treatments of the day, sulfur drugs, frequent debridement, and agonizing antiseptic washes, were failing the consulting surgeon. His face grim under the bright operating theater light, spoke quietly to the supervising nurse. "The infection is spiking. Nurse, septicemia is setting in. We've done all we can. Prepare him for amputation. It's the only way to save his life." In the 1930s, battlefield wounds rarely killed soldiers instantly. Instead, it was the aftermath, the slow, creeping rot of infection that claimed over 18% of all wounded men. A simple scratch could quickly become a death sentence, turning a minor injury into a debilitating, fatal infection within days. The problem was not the bullets. It was the bacteria. Yet, just a few hundred yards away, in the labyrinthine corridors of the Sir William Dunn School of Pathology, a solution was stirring, quite literally, in a dirty, discarded flask. Dr. Howard Florey, the tough, intensely pragmatic pathologist from Adelaide, Australia, ran the department with an iron will. He was driven, focused, and utterly lacking in the theatrical flair of men like Alexander Fleming. He was a scientist of results. Fleming, years earlier in London, had made the brilliant observation that a stray mould, Penicillium notatum, killed bacteria. It was a fascinating laboratory anomaly, dubbed Penicillin. But Fleming struggled to isolate the unstable, powerful substance and concluded it was too difficult to purify and produce in any useful quantity. He had moved on, but Florey had not. In 1938, Florey, along with his brilliant German-Jewish refugee chemist, Ernst, and a team of dedicated researchers, had decided to revisit all known antibacterial substances. When they reached Fleming's discarded mold, Florey saw not a curiosity, but a potential lifeline. Florey had stated, examining the culture plate. If this substance, this penicillin, truly attacks the bacteria without harming the white blood cells, then the problem isn't the mold. The problem is the chemistry. We must learn to harvest it. By 1939, Florey's team had successfully produced a crude, stable brown powder, the first usable extract of the mold. It was impure, difficult to manufacture, but undeniably effective in Petri dishes. As war exploded across Europe, Florey knew that their research had transitioned from academic pursuit to a desperate, critical military necessity. The fate of millions of wounded men, like Private Miller lying in the ward, now rested on their ability to turn an accidental speck of mold into a weapon against the deadliest foe of the battlefield. Infection, the race against time and against the mounting casualties, had begun. Late 1939, early 1941, the Sir William Dunn School of Pathology. Oxford. The laboratory at Oxford transformed into an industrial cottage operation, driven by a frantic, wartime necessity that overshadowed all academic propriety. Howard Florey, the quiet, relentless Australian, became less of a professor and more of an industrial foreman, obsessed with scaling up production of the precious, life-saving mold juice. The challenge was monumental. Penicillin was notoriously unstable and incredibly difficult to extract. To produce even a small therapeutic dose, the team needed massive amounts of the mold culture. The entire process was agonizingly slow and inefficient. Dr. Ernst Chain and biochemist Norman Heatley became the chemical and engineering engines of the effort. Heatley, a resourceful and ingenious scientist, realized that standard laboratory glassware was insufficient. They needed hundreds of specialized shallow containers to maximize the surface area for the Penicillium notatum mold to grow. With the war effort consuming nearly all industrial capacity, Heatley scoured Oxford, eventually improvising an entire production line using milk bottles, bedpans, and ceramic laboratory dishes. Anything that could hold the growing culture. The stench in the pathology school became notorious. It was a potent, yeasty aroma of growing mold and fermenting broth. The team, often working 16-hour days, became a tight-knit, utterly focused unit, pouring, filtering, extracting, and re-extracting the crude yellow-brown liquid that held the tiny, molecular key to defeating bacterial infection. Florey knew that small-scale production was meaningless. They needed to prove the drug worked on a human being before they could justify demanding industrial production. The first animal trials, conducted in early 1940, were stunning. 8 mice injected with a deadly dose of streptococcus were given penicillin. All 8 survived. 8 control mice, left untreated, died rapidly. The difference was stark, undeniable, and miraculous. Chain would later describe the results as a miracle, an immense drama, but human trials were the true test, and they came with immense ethical and logistical risks. The first patient was an Oxford policeman, Albert Alexander, suffering from severe septicemia that had spread from a minor facial scratch. Alexander's condition was terminal. He was already fading, covered in abscesses. On February 12, 1941, the team administered their entire, minuscule stock of penicillin, just enough for a few days. The effect was immediate and dramatic. Alexander's fever plummeted, his infection began to recede, and he showed clear, undeniable signs of recovery. Hope flooded the sterile hospital ward. But the team's meager supply of penicillin ran out before the infection was fully eradicated. In a desperate, frantic attempt to maintain the treatment, Norman Heatley was dispatched to the hospital to collect Alexander's urine, which still contained traces of the precious drug, so that Chain could extract and recycle it. But the effort was futile. Without a continuous supply, the infection surged back, and tragically, Albert Alexander died. The failure was crushing, but the evidence was overwhelming. Penicillin worked. It was not a cure for all diseases, but a specific, powerful weapon against the most common killers of the battlefield, the Staphylococci and Streptococci. The problem was no longer whether the drug was effective, but whether they could produce enough of it to save a single soldier, let alone an entire army. As Hitler's blitz raged over Britain, destroying factories and disrupting supply lines, Florey realized he could not possibly scale up production in the bomb-threatened UK. The need was too immense, the resource scarcity too great. Florey, the determined Australian, recognized that if his discovery was to save millions of Allied lives, he had to take his fragile, vital mold and his proof across the Atlantic to America. The future of battlefield medicine, and perhaps the course of the war, depended on a briefcase filled with little vials of mold. June 1941, New York City, New York, USA. The voyage across the U-boat-infested Atlantic was fraught with peril. Yet Dr. Howard Florey and Norman Heatley carried the most precious cargo in the Allied scientific arsenal, a small, ordinary briefcase containing several tiny, delicate glass culture tubes. Inside these tubes lived the Penicillium notatum mold, the biological source of the world's most potent antibacterial agent. If the submarine warfare claimed them, the global development of penicillin would be set back for years, potentially costing millions of lives. Upon arrival in the United States, Florey and Heatley, operating under immense secrecy, traveled immediately to Peoria, Illinois. This was the heart of America's industrial fermentation research, particularly at the Northern Regional Research Laboratory, NRRL, of the U.S. Department of Agriculture. Florey, focused and driven, presented his data, the undeniable evidence from the mouse trials and the fleeting success with Albert Alexander. The American scientists were initially skeptical. They had seen many supposed miracle drugs fade away. But Florey's rigorous data and Heatley's ingenious demonstration of the back extraction process, a crucial step for recycling the drug, eventually won them over. The NRRL quickly dedicated its immense research capacity to the penicillin problem. The problem, however, remained scalability. Florey's Oxford method, relying on thousands of shallow bottles and hand processing, was utterly useless for mass production. It was a matter of chemistry and biology meeting industrial engineering. Dr. Andrew Moyer, a fermentation expert at the NRRL, made the next breakthrough. He discovered that by substituting the complex Oxford broth with simple, readily available corn steep liquor, a cheap, abundant byproduct of the corn milling industry, and adding lactose, the penicillin yield increased tenfold. Furthermore, Moyer found that the mold grew exponentially better in deep fermentation tanks rather than shallow bottles, solving the fundamental industrial bottleneck. This was the turning point from a lab curiosity to a mass-producible drug. The corn steep liquor provided the perfect nutrient medium, and the deep tank fermentation technology meant that penicillin could be grown and harvested on an industrial scale. Similar to brewing beer, the vast industrial capacity of the United States, which was still officially neutral but gearing up for war production, was now brought to bear on the mold. Florey tirelessly crisscrossed the country, meeting with executives from major pharmaceutical firms, Merck, Pfizer, Squibb, and Abbott. He pleaded, explained, and demanded that they prioritize penicillin production, not as a commercial venture, but as a critical military necessity. The companies, recognizing the immense potential and responding to the looming national emergency, agreed to collaborate, sharing their research and technology, an unprecedented move in the competitive pharmaceutical industry. By late 1942, as American troops were beginning to enter the North African theater and the casualties began to mount, the first few thousand gallons of deep fermentation broth, yielding precious penicillin, were being processed. The initial doses were small, but they were now stable, pure, and ready for battlefield testing. The Australian scientists' determined journey and the subsequent American industrial mobilization had ensured that when the Allied forces finally hit the beaches and deserts, they would be armed not just with steel and gunpowder, but with a silent, biological weapon against the scourge of infection, a weapon born from an accidental observation and transformed into an industrial miracle. The ultimate test of penicillin's life-saving power was now heading directly toward the front lines. Late 1942, mid-1943, Allied field hospitals, Tunisia and Italy. The arrival of penicillin on the front line was heralded not by fanfares or press releases, but by hushed whispers in field hospitals, the quiet awe of surgeons and medics witnessing the impossible. The North African campaign, characterized by brutal tank battles, severe shrapnel wounds, and the pervasive contamination of desert sand, became the first true laboratory for the mass application of the drug. Prior to penicillin, Treating a compound fracture or a severe abdominal wound in a field hospital was often a tragic exercise in managing inevitable sepsis. The treatment for gas gangrene, a swift and deadly bacterial infection common in deep wounds, frequently involved immediate, often fatal, amputation. The prevailing medical philosophy was treat the wound, expect the infection. Captain Robert Sterling, a young American surgeon attached to the 3rd Infantry Divisions in Tunisia, remembered the despair. "We were losing too many boys," he later recalled, "not to the Germans, but to the staph and the strep. A solid leg bone could shatter, and within 48 hours, the patient was septic. You could scrub, you could debride, but the infection always won." Then, the first vials arrived, labeled simply and strictly for use in cases of severe, life-threatening infection. The initial supply was tiny, the result of the combined, yet still limited, production capacity of the American pharmaceutical giants. The distribution was controlled with military precision. Sterling's first case was a young tank crewman who had suffered deep, highly contaminated shrapnel wounds to his chest and abdomen. Pus was weeping from the wounds, and his temperature was dangerously high. He was fading fast. Sterling, following the new, tentative guidelines, began administering the pale yellow liquid. The results were immediate and astonishing. Within 24 hours, the crewman's fever broke. The angry redness around the wounds began to subside. Within days, the flow of pus ceased, and granulation tissue, the sign of healthy healing, began to form. The bacteria, the invisible killers, were simply ceasing to multiply. Word quickly spread through the medical corps. Penicillin, often nicknamed the Mould or the magic bullet, was proving particularly effective against the 2 great killers of the battlefield, staphylococcal and streptococcal infections. Surgeons found that they no longer had to rush to amputate limbs. They could use penicillin to control the infection, stabilize the patient, and then perform reconstructive surgery, days or weeks later. The impact on casualty statistics was revolutionary. Before penicillin, the mortality rate from severe open wounds and compound fractures was alarmingly high. With penicillin, the rate plummeted, particularly the death rate from septicemia. The drug performed better than any physician dared to hope, as production ramped up exponentially in the US. Moving from small batches to thousands of millions of units, the drug became standard issue for the Allied invasion of Italy. Medics carried the vials right behind the frontline troops. The cost savings alone were immense. The millions of days saved from hospital stays. The years of care avoided for maimed and septic soldiers translated into a massive reduction in the medical cost of the war, freeing up critical resources. Dr. Howard Florey's decision to transport the Mould and his data across the Atlantic had paid the ultimate dividend. The accidental discovery, guided by Australian persistence and fueled by American industrial power, was now saving thousands of lives every month. It was an asymmetric weapon against disease, and it belonged only to the Allies, becoming one of the most guarded secrets of the entire war. The true test of its power, however, awaited the single largest and bloodiest military operation in history, the invasion of Normandy. June 6, 1944, Utah Beach, Normandy, France. The beaches of Normandy were a vision of hell, a maelstrom of shell fire, machine guns, and explosions. But amidst the chaos of steel and blood, a silent, microscopic war was being fought and decisively won by the Allies. This was the war against infection, and its weapon was penicillin. The planning for Operation Overlord involved logistical nightmares that went far beyond tanks and ammunition. The medical logistics were perhaps the most critical. General Eisenhower's command knew the casualty figures would be immense, and they were determined not to repeat the fatal infections that plagued earlier wars. Penicillin was the answer. By the summer of 1944, American pharmaceutical companies, including massive operations by Pfizer and Merck, were producing the drug on a scale that was unimaginable just 3 years prior. The entire supply chain of penicillin was controlled by the military, designated as a strategic resource. Every medic, every field hospital, and every surgical team landing in France was supplied with vast quantities of the pale yellow powder, now packaged in standardized, easy-to-use vials. Sergeant Frank Davies, a combat medic with the American 4th Infantry Division, landing on Utah Beach, carried his medic bag not just with bandages and morphine, but with the precious vials of penicillin. His training was explicit. After stopping the bleeding and stabilizing the patient, the next step was an intramuscular shot of penicillin, a prophylactic measure against the ubiquitous, deadly bacteria lurking in the mud and sand. Davies encountered a young soldier with a deep shrapnel wound to his leg, a wound that, in 1918, would have almost certainly led to amputation or death. Davies applied a field dressing, administered the morphine, and then, with practiced speed, drew the clear liquid from a vial, injecting the penicillin. He didn't just treat the wound, he preemptively defeated the inevitable infection. The results throughout the Normandy campaign were staggering. The ability to stabilize severely wounded men and transport them back through the rapidly established evacuation chain. Confident that the penicillin was suppressing the onset of septicemia and gas gangrene, was revolutionary. Amputations dropped dramatically. Mortality rates from combat wounds, which historically ranged from 15% to 20%, plummeted into the low single digits. In the first 2 months alone, hundreds of thousands of doses were administered in the field. Penicillin did not kill the enemy, but it saved more Allied lives than any tank, plane, or rifle. It kept men alive long enough to recover, conserving manpower and relieving the immense social and financial burden of long-term care for septic or gangrenous patients. The man most responsible for this victory, the determined Australian pathologist, Dr. Howard Florey, received relatively little of the public credit during the war. The British government, wary of revealing the success to the enemy, maintained strict secrecy around the entire penicillin project until after 1944. Alexander Fleming, the original observer, became the public face of the discovery, while Florey, the quiet engineer of the Industrial Revolution and the man who risked his life and career to bring the mold to America, remained in the background. In 1945, the Nobel Prize in Medicine was jointly awarded to Fleming, Florey, and Chain. But Florey's enduring legacy transcends the honor. His relentless focus, his rigorous scientific process, and his crucial, determined decision to cross the Atlantic with a few vials of mold transformed an accidental laboratory curiosity into the most powerful life-saving drug in human history. The weapon that saved more lives than any other in World War II was not born of explosive or steel, but of Australian persistence, American industry, and a speck of ordinary mold.